Thank you for the introduction. And actually, I'm from University of Helsinki, and I'm going to introduce a system called MediSyn. It synthesizes multiple biomedical datasets and visualizes data uncertainties. And we have 3 research groups involved in this project, and we are all from University of Helsinki and Aalto University in Finland. First, it is a common problem in biomedical area that the datasets are dispersed in various sources, which hinders users' generation of structured knowledge. So how to integrate the biomedical datasets to facilitate human insights can be a challenging problem. For example, there are 2 datasets, the cosmic drug resistance mutations and the cancer genome interpreter, which both of the datasets represent drug-target relations. So if we have one interface which can support the user to search across the various sources, it can save a lot of user efforts. And on the other hand, there are uncertainties in curated biomedical datasets. For example, usually there are missing data in the curated biomedical datasets. And also when we integrate the multiple datasets, there can be data conflicts. So in this example, the Y253H mutation and the dasatinib shows resistant relations from the cosmic dataset, but the cancer genome interpreter shows this pair of drug and mutation is responsive to each other based on the guidelines. And the curated data are mainly collected from the publications. So it is important for the user to aware there are uncertainties exist in such kind of curated database. The research purpose is to use the interactive visualization techniques to integrate the biomedical datasets in a transparent way and expose the data uncertainties to support a more informed drug treatment selection. So this is the overview of MediSyn. The left list contains all the controllers, and the middle view currently integrates 2 datasets, and also it exposes data uncertainties like missing data or data conflicts, and the right side displays the publication sources of the user-selected data. So in the end, we will derive the design implications based on MediSyn. There are some related work trying to integrate the multiple biomedical datasets, like this one, PharmaTrack, which integrates the drug targets, bioactivities, and chemical structure. And this contour visualizes datasets in columns. So the user selection of the items in one column can highlight all the related items in other columns. And the domino uses explicit links to interrelated datasets. MediSyn is different from the related work. It uses overlaid layers to interrelate drug targets, bioactivities, and cancer treatment biomarkers. At the same time, it exposes data uncertainties. So in consideration of the scalability, we adopted a matrix-based layout. It shows the drugs in rows and the targets in columns, and the tumor types also in rows above the targets. And we encode the data parameters based on this Mackinlay's ranking. it is a visual variable ranking, indicates the accuracy of representing different types of data, like the quantitative data or ordinal data. So we prioritized the parameters from the two datasets. And the CGI dataset, which is the cancer treatment biomarkers, and DTC dataset, which is the drug target bioactivities. Because the data from CGI has a higher evidence level comparing to the data from DTC, so we prioritize, we put the data properties from CGI with a higher priority. So the first type of data are drugs, targets, and tumor types. They are nominal data, and we adopt the best visual variable, which is the position. So they consist of the matrix layouts in our visualization. And the drug target effects, which is also nominal data. So we use the second best visual variable, which is the hue, to encode this data. And the evidence level is related to this effect, which is ordinal data, and we use a combination of position, length, and saturation. So the data from CGI looks like the bars in different colors and different lengths. And then we have the bioactivity potency level data from DTC, which is also ordinal data. And we encode in a combination of position and length. We use these overlaid layers to show the data from different sources to facilitate the direct comparison. And currently in this view, it shows the data from the CGI dataset, which the green bars represent the responsive relations between the drug and target, and the red bars shows the resistant relations. So when we turn on another dataset, and it will be shown on top of the first data layer. And in this way, we can expose the data consistency states, like if there are long black bars on top of the green bars, we can see the two datasets give the consistent results about the drug target relations. Where there are long black bars on top of these red bars, it shows the two datasets give the inconsistent result. And another feature in MediSyn is it can link the user directly to the publication sources. So in this way, it indicates the data credibility. And this video shows the main features in MediSyn. So currently, it interrelates the cancer treatment biomarkers and drug target bioactivities, two datasets. And it shows drugs in rows, targets in columns, and tumor types in rows above the targets. And when the user selects a mutation, it will be added as a new column in the view. And we encode the drug target effects in hues. So the green bar shows the responsive relations between the drug and the mutation. If the user turns on another dataset, it will be shown as another layer on top of the first layer. And if the user click on the column header, it can collect all the related drugs. And if the user click on the data cell, the related publications will be shown on the right side. And if you user hover over the DTC bars, it shows the exact bioactivity value as the tooltip. And so in order to explore the two research questions, we did an evaluation with MediSyn and with 6 domain experts. The first research question is what features of MediSyn are useful and what features need to be further developed. And secondly, can MediSyn convey data conflicts, and how will user awareness of conflicts affect user trust in the curated data as well as in MediSyn? And we use the original websites of the 2 datasets as the baseline system. And all the participants completed the 2 tasks. The first one is choose the most effective drug for a group of mutations using the baseline as well as the MediSyn. And the second task is to find the conflicts between the 2 datasets using MediSyn. And for the first task, the participants needed more time with the baseline. And for both tasks, all participants got the right answer to the questions. And through the questionnaire results, we find MediSyn tends to increase choice satisfaction and lower choice difficulty. So during the first task, the users have no idea about the conflicts in the system. And so we gather the user trust in the system and the dataset. And after the second task, they find there are conflicts in MediSyn. And we gather the user trust again, and we find the user trust in MediSyn tends to drop. But most participants, they never used any of the datasets, so they are unsure about the trustworthiness of the 2 curated datasets. And we derive the design implications based on our user studies. First, the matrix view supports the drug comparison and exposes missing data. And showing datasets in overlaid layers facilitates direct comparison of data consistency states from multiple sources. And exposed data conflicts tend to lower user trust in MediSyn. And through the experts' feedback, we find the user accessibility to data provenance, like the publication sources, appears to be an important and useful feature of the system. And also more patient data, like patient clinical history or their age, gender, can be embraced to expand user knowledge and support decision. Currently, MediSyn only integrates 2 datasets, and if we have 3 datasets or we have more than 3 datasets, we think we can use MediSyn to provide an overview of the results, like what's the possibility of this drug and the mutation are responsive to each other, and what's the possibility that they have resistant relations based on the calculation across the data sources. And the user can adjust the weights of the dataset in the calculation based on user trust in the datasets. And if the user click on the data cell, it will show the details and the sources of the data on the right. That's it. Thank you for your attention.
Thank you very much for the presentation. We have time for questions. While you're making your way to the microphone, in case you do have a question, I have one. And that is, if I understood this correctly, what you told us, if when you showed issues with the data, the trust in your tool went down. Is that correct? Did I understand this correctly?
Yeah,
Okay.
if I show the conflicts.
Yeah.
Yeah.
Yeah. So, but those are conflicts in the data.
Yes, in the data.
But that meant people didn't trust you. That's an interesting finding, that people, in fact, didn't trust your tool because you just exposed the issues with the data. Do you have a thought on why they sort of blamed that on your tool rather than on the data? Or is that maybe just how you evaluated this?
Yes, because I only evaluated with 6 participants, I think the number is quite small. So I cannot draw this strong conclusion from this result. I can just say there's a tendency that the user trust in the system will decrease.
Okay.
And about the user trust in the dataset, I think this is also an interesting problem. If the user, they are familiar with the dataset, but when they find out this conflicts, how will their trust change after that can also be interesting to explore.
Okay, great. Thank you very much. If there are no more questions,